Okay, so our fourth and final category of the organic biomolecules are going to be the nucleic acids. And I also want to explain what a nucleotide is. So I have listed in your manual nucleotide and / nucleic acids. So these are the structures, for example, that are going to make up the DNA and the RNA. So the nuclear material. All right, but that's not the only thing that they're going to be used for. They're also going to, for example, you find a nucleotide in ATP. Okay, so when we look at the array of this, and you might even be looking at this big DNA structure here, and what do you notice? There is a lot of nitrogen, an exorbitant amount of nitrogen in here. So one key thing in looking at this is that the nitrogen almost in some ways can equal out to the carbons, okay? So I'm going to go through this kind of one by one, and where I want to start is actually ATP. So I'm going to set these to the side. And we're going to start with this molecule right here, okay? Now, this is what we call a nucleotide. A nucleotide is a 3-subunit structure. What we're going to have is a nitrogenous base. We're going to have a 5-carbon sugar, and we're going to have at least one phosphate group attached. So you have to have phosphate, sugar, nitrogenous base, and that is the definition of a nucleotide. So this ATP molecule is just that, a nucleotide. So a nitrogenous base, we're going to look at that a little bit, but a nitrogenous base you can think of as being the 4 or 5 different bases that are going to make up RNA or DNA, okay? So things like uracil or thymine or, you know, all those kind of different things, okay? So that's what this is. Now, I'm not going to require that you know the names of the individual ones, but here, adenosine triphosphate, the adenosine is this nitrogenous base, okay? Now, what we have here is the sugar. I said a 5-carbon sugar. You know, you've already looked at the video for the carbohydrates and the sugars. A 5-carbon sugar is from the ribose family, okay? And then over here, phosphate. We have to have at least 1, but here we have 3. That's where we get the triphosphate name, okay? If I was to take off one of these, it would be ADP, which is adenosine diphosphate. Or you can have adenosine monophosphate, which is what that would look like, okay? So, but I'm going to leave it as the ATP. And we all know that this is our currency. This is our way that we generate metabolic reactions that require energy, because this third bond is going to break and release a tremendous amount of energy to drive those reactions. Okay, so that's ATP. What we're looking at here is 2 different nitrogenous bases. Okay, so this is one component of the nucleotide. Again, nitrogenous, nitrogen, heavy with nitrogen. Okay, so these are uracil, cytosine, adenine, thymine, guanine. Okay, so all of those guys. I am not going to require that you know when you look at this, oh yeah, this is adenine or this is thymine. But what I do want you to be able to identify are the purines and the pyrimidines, okay? So those are the subcategories of nitrogenous bases. So when you look at this, this is a small molecule, smaller than this. Okay, so how are we going to know which one is a purine, which one is a pyrimidine? The way I remember it, which is kind of goofy, but this is a smaller molecule, this is a pyrimidine, and it's a long word with this short molecule. Over here, we have a larger molecule, but it has the shorter word. So this is a purine, okay? Pyrimidine, small molecule. Purine, larger molecule. And so that's to the extent that I want you to know these things. Okay, now we're going to talk a little bit here about DNA, okay? So DNA, and I know that this is a challenging to look at, and I highly encourage you that you look at it in the lab. But DNA is a double helix that is going to, each of the helical structures is going to be bound to the other one through hydrogen bonds. And so these are polymers of the nucleotides. Okay, so on the outside, which you can kind of see running up the side here, is going to be the sugar phosphate group. Okay, so this is sort of what we call the backbone or the sides of the ladder. Okay, so we see it here on one of the helical structures, and we see it here on the other. And so they're sort of just following around in that sort of circular pattern. And then in the center of that, what we have is a nitrogenous bases bound through hydrogen bonds. So here we might have, if we see a 2-ring structure, then we know that that's a purine. And we have a 1-ring structure, we know that that is going to be a pyrimidine. Smaller molecule, longer word. Big molecule, smaller word, okay? Now, it is challenging to see this, especially on video, but we do have some of these bigger structures that I want to take a look at. So I'm going to bring this to the side and pull these guys over. Okay. So what we have here is the backbone of the DNA. And so what we have is a phosphate group, sugar. And of course, if it's DNA, this is deoxyribose. Phosphate group, sugar. Phosphate group, there would be a sugar. Phosphate group, sugar. So that is this outside part of the DNA, the backbone, the sides of the ladder. Okay? And they just continue on the same exact way. And then what's going to be attached to that are the steps of the ladder, and that's going to be our nitrogenous bases. So as I pull over something that looks like this, I have some of the sugar, the deoxyribose that's attached to the nitrogenous base. And in this case, the nitrogenous base is a pyrimidine, and that is going to be bound through the hydrogen bonds to the larger nitrogenous base, which is the purine. So in this case, white, which is perhaps a little unfortunate that they put it in white because these are not actual hydrogens, but these are symbolic of hydrogen bonds between the two helical sides. Okay? So there can either be 2 hydrogen bonds, which we see in here, or when we look at this one, the 2, okay, pyrimidine, purine, and these are the hydrogen bonds, there are 3 of them. Okay, so it's just indicating which of the nitrogenous bases that they are. Okay, so again, let me reiterate that the 2 helical structures are bound together, held by hydrogen bonds between these nitrogenous bases. Okay, so you need to know the base, you need to know that these are hydrogen bonds, and then you need to be able to understand the backbone of phosphate, sugar, phosphate, sugar, phosphate, sugar, and know what the definition of the nucleotide is. One nitrogenous base, one sugar, and at least one phosphate group. That's going to do it for our video on nucleic acids and nucleotides.